Hey everyone, I'm Samuel Purdom, master's student with Dr. Avat Shekoofa here at the West Tennessee Research and Education Center. We're crop physiologists interested in studying plant water use, drought, water management, and sustainable field crop agriculture. We're here at our small plot soybean field in Jackson, where we'll be studying drought and different irrigation strategies this year to evaluate soybean physiological parameters. What we're standing in right now looks like a greenhouse, but it's actually a high-tech piece of equipment called a rainout shelter. This shelter, when pulled over our soybean fields mid-season, will allow us to simulate extreme drought conditions in the field and evaluate how different soybean varieties perform under those conditions. We also have a center pivot irrigation system and also rain-fed treatments so that we can simulate different environmental conditions and abiotic stressors in our experiments. The first thing I want to talk to you guys about is trends that we all notice in precipitation, climate, maybe even drought. As farmers or as anybody involved in agriculture, everyone has a story to tell about precipitation, maybe it's temperature, maybe it's flooding, maybe it's drought. And so we're trying to use those narratives to help develop sustainable ways to move forward. This map here shows the 2019 soybean production across the United States. Out of 350 billion bushels produced, only about 6% of these soybeans were grown under irrigation. So that means that about 95% of soybeans rely on rainfall to produce yields. Keep this map in mind for comparison with the next several maps. This is a map from September 2019 where you can see the drought severity in the southeastern United States coming over to the mid-south. Notice that these areas are overlapping from the last map. And in 2019, if you recall, we had a very wet early season, but this drought quickly developed late in the season in these areas. When you couple this with temperature and drought have an additive effect when we're talking about soil moisture and plant water use. As temperature increases, plant transpiration and metabolism increases, as well as evaporation from the soil surface. So when we're talking about increases in temperature and changes in precipitation, oftentimes those two work together to create even more extreme environmental stresses or abiotic stresses. So with all of that, what we were really looking at is how soybean plants respond to drought and heat. No talk about transpiration and plant water use would be complete without talking about stomata. I'm sure, as most of you all are aware, stomata are openings on the leaves that control how much gas is exchanged between leaves in the atmosphere. That means CO2 in, water vapor out, and also oxygen out. The stomata control all of the gas that is used for photosynthesis. The amount of water leaving the plant via transpiration is controlled by stomata. And the degree of stomatal control, how open or closed they are, depends on atmospheric demand, how hot and dry the air is, and also soil water conditions. One way to think about how stomata act is that stomata respond to soil water in this way. When stomata remain open when soil water is insufficient, roots are unable to keep up with the demand, the plant leaves become dehydrated and wilt. When stomata close or partially close under a soil water deficit, the roots are able to keep up with that demand and keep the plant hydrated or turgid. So in this slide, you can see these two graphs. This is looking at some data from a greenhouse experiment that we did. On the left, we have a drought-tolerant line, Ellis. On the right, we have a drought-sensitive line, USG 7496. To make this graph simple, on the x-axis, you can think about it as available soil water from 100% on the left to 0% on the right. On the y-axis, we have the transpiration rate of the plants. Like I said, stomata close, or when soil water begins to dry up. As you can see, Ellis begins to close its stomata at about 60% soil water, while the USG line closes at about 30% soil water. What this means is that Ellis is actually responding more quickly to the drought, saving water for later development stages like seed fill, when water in soybeans is actually much more important than during early vegetative stages. It can also be thought about in terms of wilting. So on the left, you can see slow wilting means early stomatal closure. That means leaves are remaining hydrated, there's less transpiration happening early in the season, and that water can be saved for later for the more reproductive stages. In contrast, fast wilting means that the stomata are remaining open, roots are not able to keep up with demand, soil water becomes depleted earlier, and there's less water available late in the season. We even see this happening in the field, and we developed a visual rating scale for wilting to help us identify and contrast different soybean lines. This picture was taken in the field at the same time under same soil moisture conditions. However, Ellis, the tolerant line on the right, you can see is much more green and turgid under these drought conditions than the USG line on the left. This is due to that Ellis line closing its stomata earlier, saving that water, and being able to maintain its leaves much longer into the season. Another very important aspect of drought in soybean producing regions in the U.S. is the temporary or transient nature of these water deficit stresses. Most droughts we see here are temporary in nature, lasting anywhere from a few days, a week, or even up to a month or more. As such, it's important for soybeans or plants that will tolerate these droughts to not only survive, but also be able to recover photosynthetic function, stomatal function, after the drought in order to maintain agronomically relevant yields after those droughts. So we evaluated recovery both in the greenhouse and the field. Shown above here is a plant that was under a very severe water deficit stress, almost zero soil water, zero transpiration for 4 days. And then you can see the 24-hour recovery it made on the right just by receiving recovery irrigation. So you can see that soybean recovery is significant and can really contribute to maintaining yields in the field. With that said, our main objective here is to identify soybean genotypes with drought tolerance traits that will allow plant breeders then to develop cultivars that will lead to sustainable yields. The three smaller goals that can fit under this overarching objective are to screen genotypes for early stomatal closure and delayed wilting, to screen genotypes for recovery from extreme water deficit stress, and also to develop UAV-based thermal infrared imaging techniques that will allow us to screen our genotypes in the field more efficiently in a process called high throughput phenotyping. So I mentioned a couple things there about how we're studying this. So I want to talk to you guys about how are we doing drought research in West Tennessee. I showed you earlier the rainout shelters and irrigation pivots, but we do this research in several different ways to evaluate all the physiological parameters that we can about how these plants are responding to drought. One of the ways we do this is in the greenhouse study. In our greenhouse, we grow plants in pots and apply different types of environmental stresses that range from hot and dry air to also soil drying conditions. One of the really cool methods that we use in the greenhouse is to bag the plant pots to reduce the amount of evaporation from the plants. When a lot of people talk about plant water use or water use in the field, they're talking about ET, which stands for evapotranspiration. We're taking the E out of the ET, so we're just left with T, which is transpiration, and is the amount of water that is used by the plant. By bagging the plant pots with these bags, we tie them around the stem of the plant, we are able to eliminate soil moisture or evaporation from the soil surface and from the pot. And so we know simply by weighing the pot daily how much water is being transpired by the plant. So it's a really simple but effective way at measuring the transpiration rate in real time of these plants as they dry. And that's how we developed those graphs that I showed you earlier, where we looked at the transpiration threshold for plants under soil drying. This is that same information represented in a different way. So these are a range of different varieties that have been tested in the greenhouse. On the y-axis, you can see that's the transpiration threshold. That's the level of soil water at which these genotypes are closing their stomata. You can see our drought-tolerant lines on the left are closing their stomata at a much higher level of soil water, 70 to 80%, which means that if a plant closes stomata 80%, it means there's 80% of that soil water left for use later in the season during those really important reproductive phases. However, the drought-sensitive lines on the right, you're seeing there's only 30% to 40% of soil moisture left for those lines to use later in the season. So you can see how those lines are developing wilting much more quickly and are also yielding less in the field. The same pot study we did in the greenhouse, where we were able to evaluate recovery. And so The same way we measured the transpiration rate during soil drying, we measured transpiration rate by weighing the pots during recovery. So these plants received a 4-day extreme deficit stress at 0 transpiration, and then were rewatered for 4 days. Similar results, the line Ellis, represented by the green arrow here, recovered 50% of its transpiration capacity within 4 days of rewatering, while USG 7496 only recovered about 30% of its transpiration capacity. Keeping in mind, transpiration capacity means photosynthetic capacity. Those plants that are recovering their transpiration capacity more quickly are going to be able to produce yields through because they're receiving the water they need for photosynthesis to take place. On the right, we can see the visual score, which I said earlier we rate on the scale of 0 to 5. After 4 days of recovery, Ellis, that tolerant line, had almost 0 wilting, while some of these more sensitive lines still had about 1/3 to 1/4 of leaves wilted, even after 4 days of recovery irrigation. So we saw how this experiment went in the greenhouse. Now I want to show you guys how we're going to do it in the field. Right now we're standing in our drought test plots in Jackson. These soybeans were planted about 2 weeks ago. In a couple months, they'll be receiving the rainout shelters after we drag them out into the field with tractors. So for about the first 6 weeks of these soybeans' lives, they'll receive rainfall, and then for the rest of the season, they'll be excluded from all precipitation and irrigation so we can assess how the plants are responding to those drought conditions. And you can see here in this aerial view how the field is set up. We grow these same range of soybean varieties that range from drought sensitive to drought tolerant in the field, and apply different irrigation management strategies or extreme conditions. In this case, we have an irrigated, a rain-fed, and a rain-out area where we apply our drought stress. We do the same thing at a Milan research station, with the exception of the rain-out shelters. So with these different water management schemes, we're able to see how the plants are reacting, these different lines, and then make decisions or observations about how they might be used in sustainable production under drought conditions. In the field, measuring stomatal conductance or transpiration rate is a little more complicated than in the greenhouse. So we have some high-tech equipment. We also do leaf sampling, and we also do our visual rating score on a scale of 0 to 5 as well in the field. We also have soil moisture sensors and measure soil moisture throughout the year, so we can see how those different treatments are creating different soil water deficits or soil moisture conditions. Additionally, in the rainout shelters where we had the drought in the field, we also applied recovery irrigation near the end of the season and evaluated how those plants responded as well to that recovery. This year, we will be using a UAV or drone equipped with a thermal infrared camera to image our plots in the field. This is a really exciting new aspect of our research. Then we should be able to detect differences in transpiration based on canopy temperature. This goes up the idea of the same idea of how when humans sweat evaporates and the body cools. As plants transpire, they lose that water and heat goes with it. So the more a plant transpires, the cooler its leaves will be. With this in mind, we hope to detect differences in transpiration. Those genotypes that are closing earlier in the season should be hotter with less transpiration early in the season. Then later in the season, when there's more water available for them, they should be cooler than those drought-sensitive lines. So those are the ideas we're hoping to leverage with the thermal infrared camera this year. One interesting thing to note is the rainfall that we received in 2018 versus 2019. You can see both in Jackson and Milan, July and August, we received almost twice the amount of rainfall than in 2018. So it's interesting to see how variable the climate can be when doing these experiments. All this stuff we did, what did we really accomplish? Did we accomplish anything? Well, we noticed some really big differences in the recovery ratio of stomatal conductance between Ellis again and those drought-sensitive genotypes. So we're really seeing some big differences in the stomatal conductance over the season between those different varieties. Also, last year and the years before, we've noticed big differences in yield when the soybeans are grown under the extreme drought conditions. Ellis and also its sister line continuously yield higher under the extreme drought conditions than some of those other drought-sensitive lines. So to sum that up, we are observing genetic variation in these different traits, stomatal traits like I mentioned, and also in yield. So Ellis, its sister line, and some of the other tolerant lines we're looking at consistently show higher yield and stomatal and differences in stomatal traits under these conditions. In the future, we're really excited this year to use drones and some cutting-edge technology to evaluate our soybeans. This technology is going to be very useful in making our work more efficient and enable us to use our measurements we take on the ground, called ground truthing, to correlate with this data that we're going to be receiving in the air, and hopefully pave the way for plant breeders and other crop physiologists to make plant phenotyping and screening much more efficient. With that, I'd like to thank you guys for tuning in. I'd also like to thank the Tennessee Soybean Promotion Board for supporting this project and many other projects in Tennessee, and hope to see you guys in 2 years in Milan in the field. Thanks.